Okay, welcome to the AQA Combined Science GCSE 2022 Biology Paper 2 walkthrough. Let's get to the marks. Here's question 1. This question is about the cycling of water and carbon in the ecosystems. Which reaction produces water? Tick one box. We got aerobic, anaerobic, and photosynthesis as our possible answers. Let's write out the three equations here. Glucose plus oxygen makes carbon dioxide and water. Then we have anaerobic respiration. Let's just do the animal one, which is glucose straight to lactic acid. If it was with plants, it'd be glucose to ethanol and carbon dioxide. And let's do photosynthesis. Carbon dioxide plus water produces glucose plus oxygen. Now, obviously, products are on the right-hand side of a reaction and reactants on the left-hand side. So it's a case of just look which one produces water. We can see this first equation, aerobic respiration, produces water. So there's our answer. Okay, the next part of the question, figure 1 here represents the water cycle. We got 1, 2, 3, 4, 5 labeled on this diagram. And of course, what does the question want us to do? Nice and simple. It wants us to name what each part of the diagram represents. What are the 5 processes? So number 1, we got water going up into the sky, it appears. So that is evaporation caused by heat from the sun. Then what happens to those water particles as they get up high where it gets cold? Well, we have condensation, and that is the formation of clouds. And then, of course, when it rains, you might know from geography, this is called precipitation. Number 5 is water escaping through the stomata, the little pores in leaves. That's transpiration. And number 4 is known as runoff. That's the water running through the soil into lakes and rivers. Okay, 1.3 in 2007, the population of the world was 6 billion. A study found that 4.5% of the population had severe water shortage. Calculate how many people had severe water shortage. Give your answer into standard form. Essentially, you're trying to find out what 4.5% of 6 billion is. You can do it like this, 4.5 divided by 100 times 6 billion. You're going to get 27 with a load of zeros. That's 7 zeros to be precise. Don't forget that they want the answer in standard form. That's the third mark. So it's going to be 2.7 times 10 to the power of 8. Now, there are alternative methods to finding out what 4.5% of 6 billion is. And here's one of those alternative methods, which obviously is just taking 6 billion divided by 100. That will give you 1%. So then times that by 4.5 to give you 4.5%. Again, the answer is going to be exactly the same, which is going to be 27 with a load of zeros, which is 270 million, which is 2.7 times 10 to the power of 8, if you want to put it in standard form. Why do more people have severe water shortage than in 2007? Tick 2 boxes. So we're looking at reasons why there's less water on the earth. So, really, we should say less availability of water. So, obviously, climate change increasing the area of deserts mean there's less available water. If people drink less water, that's just going to make more available water. More water is used by crops. Yeah, that sounds like the second answer. If more water is in plants, then it's not available for people. What about the sea levels rising? Well, that would make more water available. And of course, if you build desalting factories, again, that means more water is available for drinking. Leaves on a tree contain carbon. In the autumn, leaves fall to the ground. Microorganisms in the soil recycle carbon from the leaves so that the carbon is used for new plant growth. Explain how for 4 marks. This is 1.5. Let's get into it. Okay, let's see how to answer this question. You need to know your carbon cycle diagram off by heart. Really key for questions where you have to label the carbon cycle, but also for these 4 mark comprehension questions or explanations where you need to know the carbon cycle off by heart. So the first part of the carbon cycle, let's just run through this, is that carbon dioxide is absorbed by photosynthesis into plants, green plants. It's turned into glucose, obviously, and then feeding or nutrition, animals eat plants. Now, both animals and plants respire, releasing. Sorry, I got cut off there. Both animals and plants respire, releasing carbon into the atmosphere in the form of carbon dioxide. And of course, animals and plants are living things. They can die, which is death, and then they decay. Now, for a separate mark, decay is done by microorganisms, bacteria, fungi, or decomposers, they can be called. Now, they can turn dead things into fossil fuels in a process called fossilization, a little bit outside the spec. Now, they also more commonly and far more quickly, doesn't take millions of years, can do decomposition, which is where they break down organic matter and release carbon dioxide. And again, as we said, the fossil fuels can be burnt in a process called combustion with oxygen, and that produces carbon dioxide as well. So once you've learnt this carbon cycle off by heart, any question you get on it will be really easy. So all we have to do here is see that the question is asking us, we're starting from dead leaves. How does the carbon in dead leaves get back into plants to enable plant growth? So really, those dead leaves are broken down by bacteria, fungi, or microorganisms. Point scored. In the process called decomposition or respiration, they release the carbon as carbon dioxide into the atmosphere. Another point scored. The carbon dioxide in the atmosphere is taken into plants, green plants, by photosynthesis. Another point scored. It's turned into glucose. Another point scored, where it can be used to release energy. Obviously, it releases energy via respiration, but then that energy can be used for growth. Also, that glucose can be turned into amino acids and proteins, which are essential for growth as well. They do that by adding nitrates from the soil to amino acids. Those of you doing triple. But essentially, these are all the ways that that energy can end up being used for growth. So the key to any question on the carbon cycle is knowing this carbon cycle off by heart in your head, and then just fitting it to the question at hand. And you can use this diagram in your head to explain every single question on the carbon cycle. So learn this, guys. Very, very important. And there's my written answer. But let's move on to the next question and get through this walkthrough. By the way, hit the like, subscribe, and all the usual stuff. So what is one benefit of fallen leaves for living plants? Well, that'll be nitrates released into the soil. Reason being, nitrates plus glucose make amino acids and proteins for plant growth. Okay, let's move on. So the next question is question 2. And this question is about water pollution is a problem for humans and wildlife. Explain how human activities are polluting rivers, lakes, and seas. And that's 6 marks. So really the first thing I want to talk about here with water pollution, you should be thinking plastics, you should be thinking toxic chemicals, nuclear waste, fertilizers. Well, the first thing I'm going to think about is fertilizers. Now, this is my little can with some fertilizers being poured out onto soil. I know, D grade, GCSE art. So how do fertilizers actually pollute the water? Well, let's do a little diagram here of what's going down. We've got our soil here, and essentially you've got your water, like this could be a little lake. And in the lake, I've got a little fish, and we need some plants inside the water in a lake to oxygenate the lake because they do photosynthesis and release oxygen. And here's our sunlight shining down, which is key for the lake and for the photosynthesis of the plants in the lake. However, when it rains, nitrate fertilizers run into the lake and cause algae, which is a green living organism, to grow over the top of the lake. This blocks the sunlight, prevents oxygen coming out of the plants, and deoxygenates the lake, kills the fish. So really, we just want to say that fertilized fertilizers are used by humans to grow crops for growing the population and feeding the population, really. And fertilizers can run off into lakes and rivers and cause algae to grow, or algal blooms is a way of saying that. This blocks out the sunlight, stops photosynthesis, and deoxygenates the lake, killing any organisms in the lake. Of course, we shouldn't forget that microorganisms then feed on those dead animals and plants in the lake and use up any remaining oxygen, further deoxifying the lake and making it toxic. The fertilizers themselves could contain chemicals that are actually toxic to living things in the lake, and they get into the food chain. And humans, of course, transport chemicals on boats, things like crude oil, which can spill into the water, and that is toxic as well and can poison and kill animals and plants and marine organisms. Of course, humans use plastics, which are dumped into the water and landfill sites and end up getting themselves into the sea. Animals can get trapped in them, but also plastics don't decompose fully. They're non biodegradable and they can form microplastics, which get into the food chain and the ecosystem and cause harm. Let's not forget herbicides and pesticides used by humans on crops can end up in food chains and they're toxic and can cause harm. Feces and urine, human waste is often dumped into the sea. Pathogens feed on it, and these pathogens can also be harmful to the marine habitats. You could add the sulfur dioxide from power stations and automobiles. Burning of fossil fuels forms acid rain when it reacts with water in clouds to form sulfuric acid. This changes the pH of the water and can kill fish and plants in the water. There are many more possible answers for this question. You should check the mark scheme in the description, and you can find the link to the paper in the description too. So, here in this question 3.1, we're told that water is an abiotic factor, and we're asked to name one biotic factor which may affect the number of buttercups growing on a field. And biotic, well, what does that mean? Bio means living. So, we're talking about living factors that can affect populations. So you've got the students in the school, they could be walking through the field and trampling the buttercups. So definitely people walking through the area or other animals walking through the area. Then you've got competition between other plants like the grass on the field that's trying to take the water or minerals from the soil. So competition, that's another living factor. And then there's things like herbivores, rabbits. Do they go on the field and nibble on the buttercups, or insects, which might eat the buttercups as well? And you've also got pathogens which cause disease. Maybe there's specific buttercup diseases that wipe them out and reduce their population. And once again, you can check the mark scheme for all possible answers, but I would recommend only using one answer here because it does say one factor. And you don't want the examiner taking off the mark because you've given two factors that affect the population. Let's move on to 3.2, which is describe a method to investigate if the amount of water in the soil affects the number of buttercups in the field. So here we need to describe a method to investigate if the amount of water in the soil affects the number of buttercups in the field. Earlier in the stem of the question, we were told there's a dry area to the field and a wet area. So we want to split the field into two, into the dry side and the wet side of the field. And the clue here is that you're looking at investigating the number or the population size. And when that happens, you want to use the method which we use, which is random quadrating. So pick a side to start with. It could be the dry side or the wet side. Here I'm starting with the dry side. You're going to place 10 quadrats randomly. Now, how you do this, you can say shut your eyes and throw the quadrats, spin around and let go. Or you can make the field into a grid, number it, and then use the random number generator on your calculator or a piece of paper and a number out of a hat to generate the coordinate for placing the quadrant. And of course, if you're ever asked why we do random sampling when estimating populations or in ecology, it's to avoid bias. So we don't want these students to pick an area of the field that's nice and sunny or an area that they want to go to. And of course, once we've placed our quadrats randomly, what do we want to do next? Well, we need to look in the quadrats and then we need to identify the buttercups because you're going to have grass, clover, and other different species inside the quadrat. So, how are you going to identify what's buttercups, what's clover, and what's grass? Now, you can say you're going to use a key, which is a piece of paper which will have a photograph of the different plants with their names, and you follow the method on the key to identify the species. Or, of course, you could use Google and use your mobile phone, use a tablet. You need a method to identify what is a buttercup. Now, once you've identified the buttercups in a particular quadrat, one of your 10, you need to count up the number of buttercups in the quadrat. Now, if you're looking at moss or something that's hard to identify, like grass, because there's so many of them, then you use something called percentage cover, where you estimate the percentage of cover in the quadrat. Once you've added up the number of buttercups in each quadrat, calculate the mean number of buttercups for your 10 quadrats. So for example, if our quadrat is 1 meter squared and our field is 200 meters squared, then 200 quadrats fit in the field. The mean number is 7 buttercups, so 7 times 200, our estimate would be that we have 1400 buttercups in the field. This would certainly save you a lot of time rather than counting 1400 buttercups. Finally, you would just need to explain that you're going to repeat this on the other side of the field, in this case, the wet side, find out the estimation of the population of buttercups on the wet side. You could plot this in a bar graph and you could do a comparison. It's also probably important to mention how you decide what's the wet and dry side of the field. So you could do this using a water meter or a hydrometer, something that actually gives you a quantitative measurement of the moisture in both sides of the field. So you can decide which side is the dry and which side is the wet. Now, an alternative method that is laid out in the mark scheme is to use the quadrats in a transect, which is where you put them in a line along a tape measure or a piece of rope at regular intervals, and you count the number of buttercups. You start at the dry end, which you'll find using the soil moisture meter, and work towards the wet end. Now, with a transect, you present your results in something called a kite diagram, which shows the distribution of the buttercups or the population you're studying as you work your way across a specific area. You'll find the perfect answer in the mark scheme for this. Okay, let's move on to the next question, which is going to be 3.3. And we're possibly going to get into doing some data or graph drawings. Let's have a go. Question 4. This method is used. Corn plants were grown in a large box of soil. Soil nitrate ion concentration in the box was kept at 0 part per million. All the corn from each plant in the box was removed and weighed. The mean mass of the corn was calculated. Step 1 to 4 was repeated for boxes containing soil with different concentrations of nitrate ions. Give two variables. The scientists should have kept the same. So what should we have kept the same here? So when working out what to keep the same, we should first identify what the independent and dependent variable are, because those things are changing. The independent variable is the variable that the scientists are changing or that you are changing. And the dependent variable is what you're measuring, which here is the mass of corn. Of course, the concentration of the nitrate ions is the independent variable. So they don't want to keep either of those the same. So you have to think about what you want to keep the same so it doesn't affect the growth of the plant. Because you really want to find out, is it the nitrate ions that's causing the change in the concentration of the nitrate ions that's causing the plant to grow, or is it one of these other things like type of soil, the pH of soil, mineral content, mass volume of soil? Think about the limiting factors of photosynthesis like light intensity, temperature. We've also got the amount of water or the starting mass of the corn. All of these are things that could affect the dependent variable, the mass of the corn. So we need to control them. So 4.1 just asks you to give two variables the scientists should have controlled in the investigation. And I've gone for temperature and light intensity, which are two of the limiting factors of photosynthesis. Okay, let's have a look at this question. This is 4.2. Okay, 4.2, complete figure 3. You should label the y-axis, use a suitable scale for the y-axis, plot the data from table 1 and draw a line of best fit. So we've got a graph here and it's worth 4 marks. And over here we have a table of results. Now you always have the independent variable, the thing that you change or the scientists change, in the first column, and the dependent variable in the right hand column. Now normally this independent variable goes along the x-axis at the bottom, so along the bottom of the graph, and the dependent variable, the thing that you measure, and goes on the y-axis. Now you can see you're going to get 1 mark for labeling the axis because there's 4 marks here and there's 4 bullet points, so they're kind of giving you a clue. 1 mark for labeling the axis, 1 mark for a suitable scale. So as long as your graph takes up over half of the graph paper, you'll score that mark. And then 1 mark for putting the points on from over here into the correct place. You're allowed to be half a small square out, and then draw a line of best fit. So let's go. The first point was to label the axis. It's been left blank here. Now we've got soil nitrate ion concentration in ppm. So we have the independent variable on the x. We need this dependent variable on the y. So you literally type out exactly what it says there. So here we're just putting down the dependent variable, mean mass of corn per plant in grams. That scores us 1 mark. The next mark is for making the scale. So we want to identify the smallest number, which is 122, the largest number, 268. We want to make sure that our scale will go all the way up and above 268. So I'm going to go up in 40s. So 40, 80, 120, 160, 200, 240, and 280. So each tiny square of mine is worth 4. And I do that by dividing 40 by 10, and I can work out that 1 square is equal to 4. So the next mark is going to be for plotting point. So we've already scored 1 for the label, 1 for the scale, and the next mark is for plotting points. As I said, 1 square is going to be equal to 4 because 40 divided by 10 is 4. So we know that 1 baby square is worth 4. So I want to go to 0, put that first point in at 122, and then I want to go to 10, put the second point in at 190. Now, I then need to go to 20 parts per million and put one in at 256. Oh, just make sure I've got that right. Hold on a minute. That's over 260. Right. So just make sure you've got this right. Each square is worth 4. So I start at 240 and work my way up to 256. Put that in there. Sometimes you've got to use half a square, which will be 2. I'll then go to 30 parts per million and go to 268. And now I have my last 2 plots. The one for 40 is at 240, and my final plot is at 184. There we go. And that's your 3rd mark for doing the plots. Now your 4th mark is going to be for adding a line of best fit, or in this case, a curve of best fit. And they'll often tell you in the question it's a curve, and that means you've got to do a smooth line. Don't take your pencil off the paper. That's a bit crusty, but it's hard on the computer. That's a little bit better. We'll take that. So for this next question, we need to describe the relationship between soil nitrate ion concentration and the mean mass of corn per plant. We also have got to use data from the table 1 in our answer. So we can see here that as the mass increases from zero, well, as the ion concentration increases from 0 to 30 ppm, so does the mass. It goes up from 122 grams to 268 grams. So that scores us the first mark for spotting the first pattern there in the results. However, the second pattern in the results that scores the second mark is that from 30 ppm to 50 ppm, the mass decreases from 268 to 184. And so for these 2 points, you'll secure for these 2 points, you'll secure both marks. Okay, 4.4, farmers add nitrate fertilizer to fields where they grow corn. Nitrate fertilizers are expensive. Evaluate the economic and environmental implications of adding fertilizer to soil in nitrate ion concentrations ranging from 0 to 50 ppm. So you can look back at the table to see the effect of the fertilizer. And we just answered that in 4.3, that 0 to 30 was pretty good at increasing the mass, and 40 and 50 caused a decrease in mass. So we really just need to evaluate what the farmer should use, why, thinking about the economics and the environmental implications. So what I would take into account is, when answering a question, it's good to circle keywords and key parts. So evaluate generally means consider all angles. Economic and environmental means in our answer for 4 marks, we're going to need to talk about both to get 4 marks. If we just consider the monetary side to using fertilizers, we won't get all the marks. So we need to talk about environmental implications as well to get all the marks. So an increase in mass or yield. So an increase in mass or yield has to take into account the cost of the fertilizer. So you might use a fertilizer to grow more corn, but if the fertilizer costs more than the extra corn you get, not worth using. Another point is that increasing fertilizers up to 30 ppm will be more expensive, so than not using any fertilizer at all at 0 ppm. But we saw from the data it did give us a higher yield, which means better sales, more money, you can buy a Lamborghini. Another point to consider, just changed up the color here to break up the writing. Above 30 ppm, fertilizer costs even more money. So it's more expensive to buy fertilizer, we can assume, at 40 than at 30. So it costs even more money to buy, and it actually gave us a smaller yield. So what's the point? You're not going to make as much money. So you're going to get less money via sales, and also you're just wasting every extra bit of fertilizer above 30 ppm. Now, the nitrate concentration of the soil, this is something we need to consider, might already be high. So you really need to find out the nitrogen or the nitrate concentration of your soil first, because if that's already high, we might not to use an as high fertilizer, and we can therefore save money that way as well. 4, fertilizers cause runoff. We talked about this earlier. When it rains, fertilizers run into rakes, rivers and lakes, and can cause something called eutrophication. Now that's beyond the spec, eutrophication, but the description of it is essentially fertilizers can cause algae to bloom, block out the sunlight, stop photosynthesis inside the water happening from plants that live in the water, and that deoxygenates the water. So that's an environmental implication. Also, they could contaminate drinking water. You don't want to turn your tap and drink some drinking water. Oh my gosh, check that spelling, that's bad. And have it contaminated with fertilizer. And of course, they can cause just general pollution of ocean and rivers. That will get you a mark, or lakes or seas. Any water course will get you a mark. And then there's transporting fertilizers down the motorway. Costs a lot of money. And those vehicles that transport the fertilizers could be ships if they're coming from other countries, or planes. They use a lot of energy and they release greenhouse gases, those transport vehicles, which causes global warming. So there's all these environmental implications. Now, the first 4 are mainly just cost implications, and then the second 4 are environmental implications, these ones in red. You need a combination of 4 of these. You can have 3 about the cost and 1 environmental, or 3 environmental and 1 cost, but you need to have at least 1 from each section. Okay, moving on, we've got question 5 here. Blood glucose concentration in the human body needs to be kept within the normal range. Figure 4 shows that 2 hormones control blood glucose concentration. So we have a diagram here with the pancreas, which is an organ in our body. And then we have blood glucose concentration here and 3 options: low, normal, and high. So when blood glucose concentration is low, let's follow the arrow up. Something happens in the pancreas, which causes hormone A to be released, and our blood glucose concentration returns to normal. And then when the blood glucose concentration is high, we're talking about after a meal, after eating something very sugary, something happens in the pancreas. Hormone B is released, and our blood sugar returns to normal. What type of hormonal control is shown in figure 4? Well, we call this negative feedback. A negative feedback is a mechanism that returns a level within the body or a condition back to normal. So if something gets too high, it's returned to normal. If something gets too low, it's returned to normal. Temperature, your body temperature, which stays around 37 degrees, is another example of this. Your body responds in ways to return it back to normal, and that's called negative feedback. Now we're going to be asked a question 5.2. I'm not going to scroll down to it yet, but it's essentially going to say name hormone A and name hormone B. We can see here that when your blood sugar is low, hormone A is released, and this increases your blood sugar. The name of this is glucagon, and not spelt like that. Glucagon. Yeah. Let's check out the other one. When your blood sugar is high, this hormone B is released from the pancreas, and it lowers your blood sugar, and that is insulin. Awesome. 5.3 is a dirty 6 marker. So 5.3 is a 6-mark question. Explain how the 2 hormones in figure 4 keep the blood glucose concentration within the normal range for 3 hours after a meal. After a meal, your blood glucose concentration is high. The pancreas releases or secretes insulin. Insulin causes glucose to enter liver and muscle cells. Okay, insulin causes glucose to enter the liver and muscle cells. I also like to think of the word insulin as having an L in there, which stands for liver, and the word in the liver. So it puts the glucose in the liver. What happens inside the liver? Inside the liver and muscle cells, glucose is converted to glycogen. Look, am I terrible? I've not got full stops. I'm using random capitals. But yeah, so the insulin also triggers the liver and muscle cells to take the glucose and turn it into glycogen, which is a storage molecule. This lowers the blood glucose concentration back to normal. When blood glucose concentration falls below normal, which it will do eventually, then the hormone glucagon is released from the pancreas. This breaks down glycogen into glucose. The glucose enters the blood. Now, another thing here, check out the word for glucagon. Glucagon. So I think of the gon at the end of this word as it makes glucose gone from the liver. I know, stupid, but whatever we can do to make it easier to remember. This increases the blood glucose level back to normal. And we call this whole process negative feedback, the way that a condition within the body can get out of the normal range and then be returned to the normal range. It allows the body to control different levels within a very narrow range. 5.4, follicle stimulating hormone, FSH, and then luteinizing hormone, LH, can be injected into a woman to help her become pregnant by sexual intercourse. Explain how injecting FSH and LH will help a woman become pregnant. Well, essentially, we have to know, and this is really just asking you, what is the job of FSH and LH? And then you need to link it to a woman becoming pregnant. So the first thing is, do you know what FSH does? FSH ripens, matures, or develops eggs in the ovary. So if you take FSH, you're going to have more mature and more developed eggs ready to go. And LH, when it is high in the blood, causes the release of an egg. This is called ovulation. So now, how does taking FSH and LH link to becoming pregnant? Well, obviously, if you have more ripe and mature and ready eggs, and they're getting released more because of LH, you have more chance of a sperm and egg successfully meeting in fertilization. And that's why a woman will get pregnant, because there's more chance of a sperm and egg getting together. So more chance of egg and sperm fertilizing. Why? Because we've got more eggs, so more chance of collision between eggs and sperm. And there's your 3 marks. Now, 5.5, in some women, the injections of FSH and LH are the first steps for a process called in vitro fertilization, or in vitro fertilization, IVF, as it's more commonly known. Describe the remaining steps. So the woman has already been injected with FSH and LH. That means her eggs are ripe and she's released lots of eggs. So what are the remaining steps? First of all, the eggs need to be extracted. So they have to be removed from the woman. Then mixed with sperm outside the body, probably in some sort of petri dish, but they're going to be mixed with sperm for fertilization. So the eggs are going to be fertilized externally, hence the in vitro, which means outside of the body fertilization. So third step. Then we wait for embryos to develop. And any healthy embryos are chosen and implanted in the woman's uterus in a small medical procedure. So, and then finally, we need to implant it back in the uterus. So these are our keys. The egg needs to be extracted, mixed with sperm, fertilization. Then we take an embryo, we implant it in the uterus. And they're the key terms you're looking for those 4 marks. Okay, 5.6. There are 2 different processes of cell divisions in humans. Describe 3 differences between cell division to form sperm cells compared with cell division to form liver cells. Meiosis is used to form sperm cells. So sperm and egg cells, which are the gametes or sex cells, are formed during a type of cell division called meiosis. Whereas mitosis is used to form liver cells. So your liver cells are replicated and reproduced in a process called mitosis. Meiosis involves 2 cell divisions, and mitosis 1 cell division. You'd only use 3, but I'm going to give you 4. So we can say 4 daughter cells are formed compared to 2 daughter cells when a liver cell divides. So we get 2 daughter cells. God, I can't spell daughter, right? We get 2 daughter cells, or new cells, as might be more politically correct, when the liver cell divides. But when we produce gametes or sex cells, whether it's eggs or sperm, we make 4 daughter cells or 4 new cells. And then we can also say sperm cells are haploid. What does that mean? They have half the chromosomes, or 23. So you might want to say they have 23 chromosomes or half the chromosomes, whereas liver cells are diploid. And you can just leave it as diploid, but you could explain that if you wanted to say they have 46 chromosomes. So they have double the amount of chromosomes of a sperm cell. And those are your points that will finish off question 5. Awesome. Okay, question 6. The polar bear is a mammal that lives in Arctic habitats. 6.1, complete table 2 for the classification of the polar bear. Ursus maritimus. Maritimus. Oh my gosh, sounds a bit strange. Right, here we have the groups and here are the names. Now, in order to remember the kingdoms or the classification system, we have king prawn curry or fat greasy sausages. Now, this only applies to this bit here, where we can go kingdom, then we have phylum, which is my prawn in my mnemonic, then class, which is curry, order, which is or, and then fat is family, and then greasy is genus, and then sausages is species. Now, what we can also do, the kingdom is animal, or if you want to be fancy, you can say animalia, but you can just put animal. You also have above this classification system, Carl Woese in the 1990s invented the 3-domain system, and animals are eukaryotic, so they're part of the eukaryotic domain. So eukaryotic, eukaryot, eukaryotai, whatever. You can write whatever in there, okay? So we can add a new layer to our mnemonic and call it delicious king prawn curry or fat greasy sausages, and that tells you about the domain. Scientists have been measuring the area of sea ice in the Arctic since 1980. Figure 5 shows the area covered by sea ice every September. On the left here, on the y-axis, we have the area of sea ice in millions of square meters. And at the bottom here, we have the dates from 1980 all the way up to 2020 on our x-axis. Let's look at the question. So we've got a 3-mark question here, and we're just going to go through this first of all, because I'm then going to go back up to the graph to work it out. So 6.2, determine the annual rate of loss of sea ice. So the ice has been shrinking or being lost between 1985 and 2017. A trend line's been drawn for us on figure 5 to help us. It's 3 marks, rate of loss in millions of square kilometers per year. So what we need to do here is go from 1985 to 2017. So what they're testing you here is on your ability to read a graph. So 1985 is here. So what we do, if you're in your exam and you get all of this wrong, you will gain marks from drawing lines. So we're going to draw a line. I'm going to use red, so it will stand out. There's 1985. Okay. What I want to do is bring it across and read what this number is. Now it's bang smack in the middle of a small square, so I need to work out how much a small square is. Now this is 8, this is 7, and there's 10 squares in between. So each square is 0.1. So we have 7.1, 7.2, 3, 4, 5, 6, 7.5. So this is really pretty difficult, but 7.75 is what we're looking at as our number over here. Okay. So 7.75, I put it over there. Let me just highlight that for you guys. 7.75, that's my reading. Now, remember, in all mark schemes on things like graphs, you won't have to be exact, but you have to be pretty damn close. Okay, they'll have some leeway either way if you're not quite on that. So again, here we've got 5 years in between each section. So we go 2010 to 2015 or 2015 to 2020, but we got 10. So each one is half a year. And I want 2017, so I want to go 4 years over. So let's go. I want, each one is 2, so I want 4 squares. Oh, it's right at the exact bottom there. And again, that looks like it's halfway to me, but you know what? Real hard to tell there. That one is really, really difficult. So I'm absolutely positive there's leeway here. So going up the side here, remember we were going up here in point 1. So this looks like 4.3. It may be 4.35, to be honest, as it's not exact. I'd say it's 4.35. So what do we do now? Now we've got our two numbers. We need to find the range. So we need to subtract the smallest from the largest. So we're going to go 7.75 minus 4.35, and that is going to give us a total of 3.40. Superbulous. Now, what do we need to do with that? That's going to score us 2 marks. You're going to get a mark for all this working out we've done on the graph. We're going to get a mark for coming up with a number that is within the range in the mark scheme, which will probably be like 3.38 to 3.45. So anywhere within that will probably be accepted as the correct amount of the correct change in the area of the sea ice. Now they want the rate of the loss. It's gone down, so the ice has been shrinking, but they want the rate. Whenever they use the word rate, it means you have to divide by time. So we need to divide by the time. We've got 1985 going up to 2017. So again, we just need to find out how many years that is. And if you do that, you're going to find out that 17 plus 15, we have 32 years. So we need to do 3.40 divided by 32, and that is going to give us our rate. And let's see what we get there. 3.4 divided by 32. We are going to get 0.10625. 0.10625 square kilometers, or million square kilometers per year. So yeah, the unit is already written out down below, but I'll write it about again down here, million square km per year. That is the rate. Awesome. And that there is our answer for all 3 marks. Again, there's going to be a range which you can check in the mark scheme just to make sure that you've read the graph correctly. Okay, and that's where our answer would be. Down here, they've already put the unit. That's our rate of loss. And here's the next question. The total number of polar bears living on the Arctic ice is not known. The hunting of polar bears has been banned or reduced in some areas. In some populations, the average mass and height of polar bears has decreased. Polar bears eat seals. Seals live on the sea ice in the winter and raise their pups there in early spring. In the summer, seals live mainly in the sea catching fish to Yeah, that was supposed to be fish to eat. Polar bears spend much of the year hunting seals on the sea ice and in the sea nearby, so they go on the ice and in the sea. The sea ice area is at its lowest each year in September, and at the end of the summer, we're talking September, August kind of time. Polar bears feed mainly in early spring and again in autumn to build fat stores so they can survive the winter. Now, during the winter of 2017, scientists measured the metabolic rates of 9 female polar bears and found them to be much higher than expected. They found their metabolic rates to be higher than expected. Cameras attached to the female polar bears showed they had to swim a long way to find seals to eat. Okay, let's highlight some of the key points that are going to help us with this comprehension question. It's good to highlight key facts in these comprehension questions. They help you keep focused in the exam. And when you come to answering the questions, they obviously help you pick out bits that are important for your answers. So, suggest why polar bears find it harder to catch seals in the autumn than in the spring. Well, we're told in the text, and suggest means you've got to come up with your own ideas, sensible ideas. We've been told in the text that there's less sea ice in the autumn, so the polar bears have got to swim more, and that might require more energy So, one of the possibilities is that the polar bear population could decrease. And here are a number of reasons. We've got the shrinking sea ice, so a smaller habitat, smaller area for hunting seals. With less sea ice, there's going to be less seal pups around, which are easier to catch. There's going to be more swimming in order to catch, which uses a lot more energy, um, and so forth. So you've got a load of different answers here. So less food. We were also told that they had decreased in mass and height, and that all goes to the fact that they're using up more energy and less able to store those fat stores to survive the winter. Now let's look at the reasons the polar bear population could increase. So hunting seals or polar bears could be banned, meaning greater food supply or just more polar bears um, surviving. We've got greenhouse emissions. We might reverse climate change by reducing greenhouse emissions. And there's quite a few other possible answers, and you can check these on the mark scheme. The link for it is in the description.
You're still here? It's over.